In this presentation, we are going to talk about the applications of persistent homology to biomolecular data. We mainly focus on the estimation of recombination rates using topological data analysis. Why is it important to study recombination rates? Recombination is the variation in genes and genomes in organisms which causes changes in observable characteristics. It is important for studying the origins and maintenance of genetic diversity. Recombination can accelerate the rate at which adaptation occurs. The maintenance of genetic diversity in a species can promote survival during times of unpredictable environmental change. Before we jump into the applications of persistent homology to biomolecular data, we need to discuss about some biological terms. So starting with genomes. Genomes can be understood as long words consisting as a long word consisting of four nucleotide bases denoted by A, C, G, and D. The length of the word varies across different organisms. For humans, it typically contains about 3 billion bases. For plant genomes, it can be 2 times in length than that of the human genome. Genomes are not stable objects, they change over time. An error occurs when the genome of an organism is copied to produce an offspring. In this case, there can be two types of mutations. Point mutation, where a single nucleotide, say T here, gets replaced by an A. So that is an example of a point mutation, whereas it can be more drastic, where a chunk of nucleotide bases gets duplicated or lost. Next, we have phenotypes. A phenotype is more complex than a genotype space that we just saw. They often contain mRNAs like the structure with nucleotides, expressions of protein, the shape of the cell, ability to grow or replicate, susceptibility or response to an infection, size and weight of a multicellular organism are all examples of phenotypes. We can think of it as a function where G and E are the inputs and P is the output. So here certain environmental factors causes changes in the genotype, which results in a phenotype, which usually corresponds to characteristics like appearance, ways to respond to different environments, etc. Next, we discuss something that is very important to understand genomics and evolution, which is coalescence. A coalescent process is the process of tracing back lineages of a population to a common ancestor. So, in this example, we consider two individuals, the two green circles at the bottom, whose lineages are traced backwards or upwards in this case, until it meets a common point, which is the common ancestor for both of these individuals. The common ancestor is called the most recent common ancestor, or MRCA, which is represented by this orange circle. In this case, we start from the current generation, which is at t equals to 1, and trace upward 5 generations and reach the common ancestor, which is at t equals to 6. Coalescent models can be extended to include recombination events where it is not just where it is a splitting event in which an individual, rather than being a direct descent of only one single parent, it descends from two separate lineages which occurs at a rate determined by the population scale recombination rate rho. So here's an example of of, of mutation and a recombination events happening together, where if we look at the leftmost example, we see that ACGD is mutated in M1, where A is converted to T, and in the next step, M2, C is converted to G, which results in the sequence TGGT. Then we look at the rightmost example where at M4, T changes to A to get the result ACGA. Now looking at the sequence in the middle, which goes through the recombination event where two mutations M1 and M3 meet at 2, 3 for a recombination event, we see that TCGT and ACT combines to give us TCCT. Similarly, we also get the other two examples through basic mutation and recombination events. Why is TDA useful for estimating recombination events? Topological data analysis increases feasibility of the inference problem and implicitly ties genetic distances to modern models of population genetics via the coalescent. We introduce an estimator of recombination rates that uses persistent homology and is tailored to the analysis of very large genomic samples. Genetic trees look visually complex, but from a topological standpoint, a tree is a simplicial complex containing 0 simplices and 1 simplices, and it contains no loops. But simple horizontal evolution, such as cell division, can be represented by the presence of loops, computing Betti numbers or simplicial complexes by sequencing data, which we will see in some time. In general, TDA is useful to solve problems of inferring the relationship between genomic events and changes in phenotype. Understanding the shape of the data in genome spaces reveals that the way phenotypical changes arise. Then we review some basic concepts of TDA that we've seen in class. The Betti number has the same value as the n-dimensional holes. For example, in the figure, figure A, the first diagram has one connected component, so B0 is equal to 1, and it has one-dimensional hole, so B1 is equal to 1. In the second figure, there are two loops and one connected components, so we have B0 equal to 1 and B1 equals to 2. Similarly, we have some examples of simplicial complexes where the figure in the left is topologically equivalent to the one in the left, the triangle with 3 points and 3 line segments. And C is the example of a filtration where we have a number of points and If we take balls of fixed radius epsilon centered on the points at the center of the intersecting balls are connected in the simplicial complex. From the resulting complex, it is possible to extract topological features associated to the data at scale epsilon. Then we see genomic sequences. High dimensional space. A genomic sequence is a high dimensional space where the distance between the points is given by having distance between the corresponding sequences. In the absence of back mutation or recombination, the distance between the genome increases. On the other hand, when recombination occurs, the evolutionary space contains loops. The Petty number here changes from B1 equals to 0 when there were no back mutations or recombinations to B1 equal to 3 when there were some loops were formed after recombination events. Next we see what persistent homology how persistent homology is applied to genealogies in the absence and presence of recombination. The figure A is the sample where no recombination occurs, and we have the corresponding dimension 0 barcode diagram for sequences at the terminals. The figure B is the sequence of complexes for increasing the distance parameter choices on a set of arbitrary data. Change in Petty number due to recombination. The number of loops of the evolutionary space is related to the number of recombination events in the sample history. The persistent first Petty number of a sample to infer the amounts of recombination in the sample history. It is used to build an estimator of recombination. The estimator does not rely on genetic linkage, but rather on the topology of spaces formed by genomic sequences. To summarize this, we can say that the recombination rate increases as the first Petty number increases. Next, we see the same expressed in the form of graphs, which is persistent homology estimator of recombination, where we see that in the y-axis we have the estimated first Petty number, which is plotted as a function of the recombination rate expressed in terms of effective population size and effective. And the sample size here is n. We see that estimated Petty number is directly proportional to the recombination rate, and the second image on the right is the zoomed in image of the first one to give a clearer picture. Then we see few more graphs related to the recombination rate. Dependence of Hudson-Kaplan, Myers-Griffiths, and B1 summaries of recombination on the recombination rate at a fixed number of segregating sites. Each plot is based on 4,000 coalescent simulations of a sample of 160 sequences. The color bands represent the interdecile range, and the central lines correspond to the mean. A squared Pearson's correlation coefficient is shown in each case. Fisher information for each of these three summaries in B as a function of the recombination rate. Information was computed in increments of 12.5 per n effective centimorgans. A smoother trend is plotted by averaging windows of 101 computed values weighted by the number of simulations. Evaluating the utility of B1 as a summary of recombination. We compared it to other summaries of recombination. Specifically, we considered the lower bounds on the minimum number of recombination events of a sample history developed in Hudson and Kaplan and Myers and Griffiths. We refer to those as RHK and RMG respectively. We find that B1 has better sensitivity to higher recombination rates and it does not saturate as early as the other summaries of recombination. This conclusion was also confirmed by comparing Fisher information of the three summaries as a function of the recombination rate. To investigate whether saturation of B1 occurs in practical applications, we assumed a mutation rate of 10 to the power minus 8 mutations per base per generation and an effective population size of N effective equal to 25,000 individuals. With these assumptions, saturation effects for a sample of 200 sequences became important at genetic map distances above 0.2 centimorgans. Taking results together, we concluded that B1 is a robust summary of recombination at fine scales, where the number of segregating sites is small. The pipeline for analyzing genomic data using persistent homology. Starting from a sample of sequences, one can compute distances reflecting the similarity between organisms. These distances provide a finite metric space that in some cases can be summarized by a phylogenetic tree whose leaves correspond to the points in the metric space. Distances between branches can be estimated by the addition of weights along the shortest path. In simpler terms, we can say that the pipeline for analyzing genomic data using persistent homology is given in the diagram where data is the sample whose sequences namely S1, S2, S3, and S4. We compute the Hamming distance for each of these sequences with respect to every other sequence and create a distance matrix. For example, the distance between S1 and S2 is 3 because we need 3 mutations or 3 changes in S1 to get S2. Similarly, we get the distance matrix for all of these combinations, and then we use those and fit it using the tree algorithm to get the phylogenetic tree, which has all the sequences with the Hamming distances. Next, we have persistent homology and what we studied so far. So the persistent homology detects historical recombination events from population genetic data. In figure A there are 5 genetic sequences sampled today, represented by yellow circles developed in a single common ancestor through clonal evolution. The solid blue lines are the ones representing the clonal evolution, and recombination represented by the red dotted lines. In figure B, we see the same sample within a larger sample of the population. Persistent homology is applied to the larger sample of three filtrations shown in the figure C, D, and E. In the figure F, we see that the resulting barcode. The dimension 1 bar near the center of the figure F identifies a recombination event involving the five highlighted sequences in yellow. So this was a um, high level overview of the applications of persistent homology to biomolecular data. Thank you for listening to the presentation, and I'm looking forward to your feedback. Thank you.